Good afternoon. Thank you for joining us for the February Pests in Progress webinar. My name is Jackie Pohl, and I'm the communications person for the North Central IPM Center. We are one of 4 regional IPM centers supported by USDA NIFA's Crop Protection and Pest Management Regional Coordination Program. Our mission is to coordinate IPM across the region by sharing and promoting IPM research, extension, and teaching activities. If you have a question today, be sure to type it in the Q&A at the bottom of your Zoom screen. Questions will be answered at the end of the presentation. Our speaker today is Dr. Caitlin Rering, research chemist with the USDA ARS, and she will be presenting Pollinator-Safe Fungicides, Floral Microbial Biocontrol of Anthracnose in Blueberry. Thank you for joining us today, Caitlin.
Well, thank you, and good afternoon, everyone. Thanks for the invitation to talk. So yeah, today I'm going to be sharing some of the very recent results from my lab's efforts to develop floral microbial biocontrol products that can be used to prevent the disease anthracnose in blueberry. This project is very fresh. In fact, it's so fresh that this is my first time publicly disclosing this research. So I'll be very interested to hear your feedback, and I'll try to leave plenty of time at the end for questions. So let's set the stage by first introducing the pathogen that we're trying to protect against, and that is Pollinator-Trichum. This is a fungus that causes the disease anthracnose in many, many different plants. So pictured here are just a few crops that are suffering from anthracnose disease, but that is by far not a exhaustive summary of all the plants that are impacted. And because of Pollinator-Trichum's prolific pathogenicity, it has been sort of recognized as one of the top 10 fungal pathogens of plants. So, of course, today we're focusing on just one crop, and that is blueberry. In blueberry, anthracnose is a very serious economic threat. In fact, across the entire United States, all of the different blueberry growing regions. It primarily affects blueberry as a post-harvest disease, sometimes causing complete loss of harvested blueberry crops. Of course, for farmers, right, this is really devastating. At a post-harvest disease, a crop loss means that the farmer has already invested all of the time in sort of growing and cultivating and harvesting the fruit, only for them to rot right before they can be sold and they kind of recoup their investment. And I have a picture here of anthracnose-infected fruit. You can see, especially when they're incubated under high humidity, they get these sort of fascinating little orange oozy droplets that are just filled with spores oozing out of the tissues. So although, as I mentioned, anthracnose is associated with post-harvest losses, infection occurs way, way, way beforehand, months beforehand. So Pollinator-Trichum spores actually overwinter on dormant buds. And then in spring, overhead irrigation or rain splashing will move the spores from the buds to flowers. And then once spores make contact with flowers, it's kind of all over. Flowers will usually quickly succumb to infection in like under 8 hours. However, Pollinator-Trichum infection does not cause the flowers to kind of immediately die off or otherwise interrupt blueberry maturation process. The disease continues to progress throughout the spring, right up until blueberries ripen. And then all of a sudden, we get rot symptoms occurring. And so for this reason, in blueberry, anthracnose is also commonly referred to as ripe rot, right? That name, I think, makes sense. Very descriptive. So yeah, just to drive this point home further, I thought this was a nice figure that sort of depicts how important flowering is as the sort of vulnerable time for infection in blueberry. This is a nice figure, and I put here in red the area where there is a primary infection risk for for anthracnose fruit rot. So because infections are impossible to detect until it is way too late to do anything about them, farmers rely on preventative application of synthetic fungicides for control of this disease. And I picked up this table from just a recommendation guide from an extension office. And you can see here they recommend very early in the bloom that fungicides begin to be applied as short as weekly intervals. And frequent and aggressive sort of use of synthetic fungicides like this, while it can be very effective, it does also have drawbacks. And I think many of us will be very familiar with these drawbacks because they occur anytime we rely on any pesticide. But just to quickly get through them, heavy reliance on fungicides, of course, can promote pathogen resistance to fungicides. This is already being documented as occurring for this disease in blueberry. We also have other consequences like environmental contamination, which comes with it risk to non-target organisms, very high exposure risk to farm workers, yucky chemical contamination of food that consumers kind of don't like very much. And these chemicals are also very expensive to purchase and very expensive to frequently apply to protect the crop. The main sort of difference that I want to drive home is the sort of added consequence that is layered on when we apply fungicides to flowers. And that is namely the direct harm that fungicides have on bees. So fungicides are not acutely toxic to bees in the way that, for example, insecticides are. However, they do have sublethal toxic effects, right? So they don't immediately kill the bees, but they do harm them. So let's get into that a little bit more. These toxic effects can include a reduction in the consumption of tainted food, which can kind of render bees into kind of a starvation state. It can also increase their susceptibility to disease, like European foul brood, which is a bacterial disease, sac brood virus, which of course is a virus, and other parasites like Nosema ceranae and others. In solitary bees, fungicide exposure can interfere with their ability to recognize their nests. Very concerningly, certain fungicides actually amplifies bees' sensitivity to insecticide and miticide exposure. So they synergize these insecticides and make them even more toxic to bees. And finally, we see that fungicide exposure, it's just generally found to be associated with increased mortality rates in both adult honeybees and their larvae. And so these negative effects on bees are particularly salient for blueberry growers because blueberry is a pollinator-dependent crop. So this image shows the really dramatic effect that poor pollination can have on berry harvests. And farmers know this. They know that they need pollinators, and most farmers at least, invest significant money into stocking bees during their bloom. But of course, by applying fungicides, farmers are kind of unwittingly harming the pollinators that they actually need for good crop production. Just to really drive home this point some more, there has been for several years now discussion among beekeepers and researchers that have noticed that honeybee colonies that are deployed to pollinate highbush blueberry seem to suffer poor health after returning from pollinating those fields. And there have been a bunch of different, postulated potential causes. There's been thought that maybe it's European foul brood, maybe it's fungicide exposure, maybe it's poor nutrition, very possibly it's interacting effects. And there has been some extremely fascinating work that's was recently published, sort of investigating some of these underlying causes and trying to sort of tease apart mechanisms. If you're at all interested in this topic, I hope that you'll go and read these papers because they're really, really cool. So the first study I want to highlight here found that when honeybees are placed near blueberry fields, which I'm showing here in red, this is their figure, which is shown here in red, that they're really not able to fight off sac brood virus infection, as well as honeybees that are placed farther away from blueberry pollination fields. And if you'll remember a few slides ago, I mentioned that fungicide exposure can render bees more susceptible to higher sac brood virus levels. So that's interesting. And in another similar study, well, not similar, but sort of complementary study that examined different stressors that affect honeybees, high bush blueberry here in blue was among the highest for both total number of stressors and sort of complexity of the stressor interaction network. And really in both these studies, the authors mention and discuss fungicide exposure as a potential factor that may be driving the poor health outcomes that we see for honeybees who pollinate blueberry, at least in part. So with all that, I hope I've convinced you that fungicide use in blueberry is at very best risky for bees. So if it's so bad, why do farmers do it? Well, of course, put very simply, it's because these chemicals are very effective, and especially when they're used responsibly. Not only can they kind of prevent disease, but they can also cure it. So this leaves growers in my opinion, in a bit of a double bind. They must choose between either sort of potentially losing their entire harvest to a disease or sort of slowly contributing to declining bee populations potentially, which will in turn leave them with these stunted green berries, right? And so neither of these options is really acceptable, and we need an alternative. So let's pivot now to non-chemical control methods that are sort of currently available. In my digging, and I probably have not captured everything here, so please let me know if what I'm missing. There are very few products that are specifically recommended for anthracnose control on blueberry flowers. And of these, only one is a microbial biocontrol product, and that is Botector. Botector is formulated with a plant-associated yeast, Oreobasidium pullulans, and those of you who are kind of familiar with microbial biocontrol will definitely recognize this yeast. It's used in a variety of different products and different crops to fight different diseases. It's a really fascinating and powerful microbe. So, benefits associated with alternative microbial biocontrol strategies. These include reduced risk to farm workers, to pollinators, and to the environment. These products are often, but not always, cheaper. They don't leave chemical residues in the food, and there is very little to essentially no risk of resistance development for microbial biocontrol products. So, what are the drawbacks? Well, frankly, these products do not work as well as chemical control. Most microbial biocontrol products can provide suppression, but not total elimination of disease, and they can prevent, but not cure an infection. And sometimes the sort of limited efficacy of microbial biocontrol products can be caused because they are really not able to colonize their intended applied habitat as we might hope. So this has been observed for example, soil microbes that we spray onto flowers. Oftentimes they don't grow very well. And in other cases, some sort of non-flower specialist microbes give kind of yucky cues to bees. And so we see repellent effects on pollinators, and there's been some sort of implications that they could potentially reduce yields and reduce pollination services. And this has been documented here with Oreobasidium pullulans specifically. Oreobasidium has also been reported to exacerbate some fruit defects like russeting in pear and apple, where you get the sort of thick, corky texture on the fruit surface. And in very severe cases, this russeting makes the fruit unmarketable. And some of the recent testing just this year, my awesome technician, Oriana, noticed that spraying Botector or Oreobasidium on blueberry blooms seems to cause minor but very consistent sort of burns on blueberry flowers. So we need to do more research into that and study that a bit more rigorously, but I think that's interesting to note. So what sets apart sort of our approach to microbial biocontrol product from a more traditional discovery pipeline? Well, chiefly, we have sourced all of our candidate species directly from blueberry flowers. So in 2020, we isolated and identified the microbes that are living in blooms of both cultivated plants growing in organic and conventional farms, as well as wild native shiny blueberry plants, Vaccinium myrsinides, that are growing in nature preserves. And through that effort, we found some really interesting differences in floral microbial communities. I don't have time to get into that today, but if you're curious, you can check out our paper, which we published last year on the topic. So why is it so good to use floral microbes? Why is this a good idea? First, we have pre-selected microbes that have really already proven themselves to be highly effective colonizers of flowers, right? So there shouldn't be an issue with survival when we apply these microbes to blooms. And another sort of encouraging lesson from the floral microbial ecology literature is that floral microbes readily disperse to new flowers by hitching a ride on pollinators or other floral visitors. So they should be able to kind of quickly establish in a field and actually persist for a while, moving between blooms. And we know that floral microbial community assembly generally follows this sort of first come, first serve rule. So that means that the first species that's sort of well equipped to live in a flower that shows up is able to rapidly take over and prevent any later coming species from taking root. And so that's right. This is exactly what we might hope to see with potentially seeding beneficial microbes in that can prevent the pathogen from infecting. And finally, because these microbes are already present in agricultural and ecological sort of plant pollinator systems, they're already present in farms, they're already present in nature, they're apparently living, as far as we can tell, in perfect harmony, if you will, with both their host plants and their the pollinators. So we can anticipate or hypothesize at least that issues with pollination or damage to fruit development are probably not likely. So our efforts to survey the floral microbe of blueberries gave us a bunch of different candidate species, and then we set out to sort of narrow down this list to try to figure out which are our best bets. So we used 2 laboratory assays that are meant to model interactions occurring in flowers to sort of narrow these down. In this first assay, we tried to design kind of a model nectar. So we designed a nectar recipe that sort of mimics the amino acid and sugar concentration of blueberry nectar, and then created monocultures of each of our 14 species that we tested in that synthetic nectar. And then after 1 day, we added spores of our pathogen. And then finally, on the second day, we compared the number of pathogen cells in a control solution where there was no biocontrol species added on day 0 versus our monoculture counts. And so any sort of reduction in pathogen density would suggest inhibition in this assay. So here are our results. Out of the 14 monocultures we tested, we found 3 species reduced colicotrichum growth. And these are 2 Neocomogatea bacteria species and a yeast, a nectar yeast, Mechinacobia rancensis. And interestingly, all 3 of these species are, certainly, yes, they are present in blueberry flowers, but they are actually globally distributed. And they're found in a ton of different plant species, the blooms of all sorts of different plants. The next assay that we used is a very traditional microbial competition assay. This is a plate inhibition assay. It's been around for hundreds of years. And here we place a plug of our pathogen in the center of a plate, and then we placed 2 identical plugs of the same competitor floral microbe species on either side. And then we measured radial growth of the pathogen in the plates, and any sort of reduction. You can see here we've got kind of a football shape going on as opposed to a circle. We call that inhibition. So here are those results. In this assay, actually a majority of the species were able to at least somewhat reduce colicotrichum growth. I've highlighted in yellow all of the 8 inhibitory species. And these pink arrows are here to sort of orient you that some of the species of those species here that we also saw inhibition in nectar. So our yeast was not effective in the plate assay, but our 2 bacteria actually were effective in both of our lab assays. And we also noticed this other yeast, Symmetrospora symmetrica, which was very frequently detected in blueberry nectar, showed very strong inhibition. So based on these results, we selected these 4 microbes for further study, both because they were all frequently detected in flowers. So here I am sort of showing their detection frequency in our survey results, and because of their efficacy in one or both of our assays. However, we've quickly found, after starting to sort of move these our consortium to real flowers, that specifically Symmetrospora symmetrica, I call it SIM because I'm not great with the Latin names. It actually seems to also damage flower tissues when we apply it to flowers. So I haven't given up on SIM. I'm still hold off hope that it could be useful for a biocontrol product, but for now, we backed away from applying it to blooms. So for the rest of the talk, when I say our biocontrol species, I'm talking about the 3 floral specialists here listed in yellow. Very good. So with these lab results in hand, we wanted to test our microbes in real plants, right? And this is a critical step because, of course, many strategies, techniques work very well in the lab, but then don't translate to the field. And recognizing that it might be difficult to convince farmers to let me experiment with their livelihood, we actually set out to build our own little mini farm here on the USDA campus in Gainesville, Florida. So my technician and I constructed this field ourselves in fall of 2023 with 162 1-gallon farthing southern high bush plants. They are here, they're very small. Maybe if you squint, you can see them. I swear there are plants there. They're looking slightly bigger this year. But yeah. So here is a sort of aerial map of our field, and I'm showing each plant as a little circle. We divided the field into 4 blocks and applied our treatment. And kind of the main focus that we're going to be talking about here are the green, which are sort of microbe protected plants that were also inoculated with the pathogen, and then pink, which were only sprayed with sterile buffer and then inoculated with the pathogen. And importantly, because we kind of had a feeling that our microbes might move around the field, we chose to try to exclude pollinators by bagging on each plant, we bagged at least 1 branch of blooms. And you can see bees find a way. This is actually a flower that had fallen off its nest, and the bee is sipping a little bit of nectar through the bag that was available there. Okay, so like with the lab tests, we are testing for preventative and suppressive protection. So we are first inoculating our flowers with our 3 biocontrol species, and we did this just by suspending the cells in a sterile buffer and spray applying to the blooms. And then the next day, we hand inoculate with a pipette the pathogen spores to each bloom. And then we wait a while for berries to harvest or to ripen, excuse me. We harvest them and we incubate them in the lab and just sort of repeatedly observe them for these characteristic anthracnose symptoms. So we were very interested in monitoring the dispersal and movement of these microbes in the field. So both before spraying and after spraying, we did a very simple sort of just cutting flowers and plating to selective media to observe the sort of frequency of any colonies that are consistent with our biocontrol species. And 1 week prior to the spray, we found at least one of our 3 species were present in about 1/3 of flowers at generally pretty low concentrations. This was normal. This is kind of what we expect to see. When we surveyed again, 2 and 9 days after spraying, we found that, as we kind of predicted, we really radically shifted our floral crop microbiome here. Even a week after that first spray, or more than a week, 9 days, nearly 90% of the flowers contained one of these species. And we also saw very sharp increases in the abundance relative to before the spray as well. So this is great. It sort of helps provide evidence that as we hypothesized, these microbes will disperse, they will maintain in the field, and we can kind of shape the floral microbiome in a lasting way. However, from a scientific perspective, this was a little bit challenging because, we don't have any true control flowers at this point. The microbes are everywhere. So it was very good that we had bagged flowers that we presumably believe maintained our treatments. And the data I'm about to show you right now is all from those bagged branches where treatments were preserved. So when we compare infection levels in berries that were collected from insect exclusion bags, we see that we actually cut the number of infected fruit in about half by using the Floral Microbe Consortium. So this, honestly, I was so surprised. I really thought, probably wouldn't work the first year. So this is really exciting and gratifying. And it was also kind of interesting, I think a little bit reassuring, that we see kind of similar inhibition in the field as we did in the lab, right? In most cases, we're inhibiting the pathogen by about 50%, and that's what we're seeing also in the field. We also didn't notice any issues with the fruit that developed from our biocontrol treated flowers. They had comparable weights to other plants. Remember, these are pollinator excluded treatments, so difficult to sort of interpret this too far, but we at least have no evidence that it was obviously a problem. And I can tell you, I ate blueberries from all of these plants, and they were all perfectly delicious. We didn't notice any wounding of the tissues or anything like that. We were curious to know kind of what happens to the microbes after we deploy them in the field. So we did a little bit of work to kind of try to trace them and see where we found them or didn't find them in the field. We found them occurring a lot in surrounding wildflowers in our field, as might be expected. We also found that floral microbial communities did start to kind of revert back to equilibrium over time. So I think that's encouraging. We don't necessarily want to permanently skew the microbiome of our crop or the environment, right? We just kind of want to briefly manipulate it in a more beneficial direction and then have it revert back to normal. When we surveyed the provisions of our bumblebees and honeybees in their nests and their hives, we did not find any living biocontrol species. So that's good, I think. We also didn't find any of these species surviving in the ripe berries themselves, which I think is also good because we don't need to be concerned, that maybe these might have impacts on bees' nutrition or humans' nutrition. So you have captured me all in the middle of our 2025 field season. This picture was taken just a few days ago by my technician, Brian Smith. And we're doing a few things differently this year. So we're actually using cages for every single plant in a treatment to try to preserve our treatments a little bit more effectively and a little bit more easily. We're also adding in a treatment, comparing our microbial consortium with and its ability to prevent this disease with that of Botector, which I'm excited for. And we're going to be looking at pollinator responses in plants this year as well. That will be really important for this pollinator-dependent crop, of course. And also, we are hoping, if the stars align, to also expand our testing to another related vaccinium species, which is cranberry, which also suffers from anthracnose and also is pollinator-dependent. So stay tuned for that. And then beyond, I hope maybe in 2026 to begin recruiting farmers who might be willing to let us sort of try this technology out on their field. And we're hoping to do some more formal and rigorous testing of these microbes' effects on bee health. So as I start to wrap up here, I'm really excited about this project. I hope I've convinced all of you of its promise. This technology was recently protected by a provisional patent filed by the USDA. I think that these floral microbial biocontrol products could be useful for farmers practicing all sorts of different types of agriculture. For conventional farmers, I think there is an opportunity for them to sort of pick up these tools and use them just in the bloom. And then after the bloom is over and sort of the risk period for bees has passed, they can go back to using synthetic fungicides. And of course, for organic farmers where synthetic fungicides are not commercially available, just having another product could be great. I also think it would be really interesting to see, what if we combined Botector and Oreo basidium with our consortium? Are we able to elevate protection even more? So a lot of promise there. And I also wanted to mention, if you're interested in this topic, if you're thinking about, well, that's cool for blueberry, but I'm not interested in blueberry. I want to point you to some of the work of my really amazing colleagues who are kind of developing similar technologies for other crop pathogen systems, like fire blight in apple and pear, and brown rot blossom blight in almond. And that's Drs. Amber Crowley-Gall, Dr. Robert Schaefer, and Dr. Rachel Bennett. Okay, and finally, to close out, I want to thank the amazing scientists who I have the honor to work with in my laboratory. Thanks also to the USDA's Office of Technology Transfer, who funded us and gave us the support that we needed to actually build our little mini farm. And thanks to all of you for your attention.
Thank you so much, Dr. Rering. That was really interesting. I love learning about biological options and how they can help us expand our control options beyond pesticides. So this is really great. Just a reminder to everyone that we have a Q&A section at the bottom of the screen. You're welcome to submit questions for us to answer. And thanks for your comment, Mackenzie. I had one question. I know you've been doing this work in Florida. Is there the potential to expand this work and perhaps use similar control methods in Michigan or California?
Yes, yeah, I think so. of course, it remains to be seen. We'll have to prove it first, but as I mentioned, our floral microbial species are globally distributed, and they're found in a ton of different crops. So I'd love to test that. If anybody out there wants to partner up, please let me know. But yeah.
Great. I also was curious, do the microbes survive all the way through the mature berries phase of the crops?
No, they tend not to, and that's the norm for these sort of floral specialist microbes. They're really only found in flowers or occasionally like in really closely associated habitats like on a bee or something like that. So we don't see them tending to persist throughout the berry just development, for example, which is generally a good thing, I think. Got into.
Okay. We've got a question in the Q&A. It's just saying grower contact. If you know what he means, take it away. Otherwise, ask for more clarification.
Yeah, I'm not totally sure what that question means. Maybe they're asking if I am in contact with growers. I certainly try to be. I'm a member of the Florida Blueberry Growers Association. I regularly attend the in-person meetings and chat with people. I think it's really important to talk to growers and to get their input and to kind of make sure what we're doing over here in the lab is actually helpful and useful.
Yeah, that makes sense. Okay, well, I think we will go ahead and move on. I'm not seeing any more questions showing up. So I just want to remind everyone that our next webinar will be in April. It will be Wednesday, April 16th at 1:30 Eastern and 12:30 Central. The title of the presentation is Use of Drones for Disease Management, and the presenter will be Dr. Darcy Tolinko. So thank you very much for everyone to join us today, and thank you to Caitlin. Bye.